Excellent. Thank you, Edward. And thank you all for joining us this morning. My name is Ted Tenthoff, and I'm a senior biotech analyst, managing director at Piper Jaffray. And Piper Jaffray is, again, very pleased, very honored to be co-hosting this event this year. It's really great to see how much the event has grown over the last 3 years. And I would like to start by echoing Edward's comments and thanking the organizers and the other sponsors. I know ARM puts a lot of work into this event, and I think it's really showing how much the space has grown by the attendance here this year. So I have the pleasure of beginning our discussion today with two leaders in the field, Drs. Malcolm Brenner and also Olivier Danos. To my far left, Dr. Brenner is the founding director of the Center for Cell and Gene Therapy at Baylor College of Medicine, Texas Children's Hospital, and Methodist Hospital. Dr. Brenner received his Doctor of Medicine and interned at Charing Cross and Westminster Medical School. He conducted his residency at Hammersmith Hospital in England and fellowships at Chelsea and Westminster Hospital, the University of Chicago Pritzker School of Medicine and Immunology, and also Royal Brompton Hospital in London in hematology oncology. Dr. Brenner, it's a pleasure to get to meet you in person this morning, and thanks very much for being with us today. And next to me is Olivier Danos, who recently joined Biogen as the Senior Vice President of Cell and Gene Therapy just last September. Prior, Olivier headed up gene therapy efforts at Cadman, which is a private biotechnology company located here in Manhattan. And showing that Olivier can really be in two places at one time, he simultaneously served as the Director of Gene Therapy Consortium for the University College of London and also led a gene therapy research team at the Children's Hospital in Paris. From 1997 through 2006, he served as the Chief Scientific Officer for Genethon, which I think many of you know as a research institute that was initially funded by the French Muscular Dystrophy Foundation, as well as the Centre National pour la Recherche Scientifique in France. And Olivier served as the Senior Director of Research at Somatics, which, as Edward mentioned, was one of the early gene therapy companies that I believe was acquired by Cell Genesis. Olivier is also a founding member and the former president of the European Society for Gene and Cell Therapy, and he received his master's in genetics and molecular biology at the University of Paris, Orsay, and a Ph.D. in biology at the Pasteur Institute. So bienvenue, Olivier. Yep. So last year at this event, I think I said that a revolution was quietly taking place, and over the last 12 months, someone's clearly turned up the volume. We have witnessed what I would say is an explosion in gene, cell, and regenerative therapies. We're now on the verge of the first approvals that could be forthcoming, multiple programs in man, some even with clinical validation. Importantly, the progress is starting to be recognized by industry in terms of partnerships and even acquisitions, as well as Wall Street, both in terms of the number of IPOs in the space and also the impressive price performance that we've seen from the sector. And I want to start off with a few questions myself, but I would like to keep this interaction. So if anyone has any questions, don't hesitate raising your hand, and we'll try to get those in. So Olivier and Malcolm, what do you think is really leading fundamentally to all of this success that's taking place? Why are things starting to happen now?
You can. Okay, well, I'm not absolutely sure it's starting now. It's getting visible now. It's been a lot of activity over the past 25 years with ups and downs. What we're seeing now are actually a lot of enthusiasm based on facts, which is the good thing, right? It's not only based on whatever hypothesis you can make or ideas you can sell. It's really the technology for gene transfer, gene therapy, and cell therapy. Those technologies have now reached a point where one can really make demonstrations of efficacy, proof of biology in the clinic, in patient and clinical trials. And there's been an accumulation of those over the past, let's say, 10 years. That includes new technologies. We can go back on that. But basically, that led to, and there's a number of triggers. There's this accumulation of positive data, sometimes in small, very well thought out clinical trials, but really good data. and that have led, at least today, in only one case, in the approval of a drug, right? Talking about Glybera, that I guess everybody knows about, which is, it's not going to be a blockbuster, but I think it's a landmark, it's an important milestone because it appears now on the screen of people somewhere else, right? Gene therapy is a product. So that's, I don't know, there's probably some psychological effect associated to that, but it becomes true, right? So the interest shifts from something that was, yeah, a possibility, something that had been very oversold in the past, lots of hype at the wrong time. So it becomes now something which is true.
Real. That's real, exactly. And Malcolm, you're doing things a little bit differently down at Baylor and Texas Children's, but what do you see that's kind of changed and that's leading to all this excitement right now?
Yeah, it is very interesting, and I think actually quite minimal changes have occurred. I think what we've seen is a kind of sociological rather than a scientific phenomenon. It's the tipping point that's always referred to. And I think people have accumulated successes over the past 20 years. There's been a huge investment, not so much from companies, but from NIH, foundations, and so on. I mean, we've received, over the past 15 years, I added it up, almost $300 million in grant funding, and that's just one center. And that's a lot of money to put into treating patients and getting successes. And eventually, it reaches a level where people sit up, pay attention, and believe in it. And I think the other factor that really helped that, honestly, was the success of the functional antibodies, the PD-1 and the CTLA-4, where people suddenly realize, oh yes, the immune system actually can start eradicating cancers and actually can be very powerful.
How much is this a recognition of the sequencing of the genome back in 2002? Was that one of the initial triggers, kind of the starting point of the race to provide us with this genetic information that is now helping us understand some of these targeted conditions? Certainly, Olivier, and some of the things that you're doing, but Dr. Brenner as well with some of the stuff that you're doing too. Is that one of the pieces of the puzzle that sort of fit into place, do you think? Are we seeing a recognition of that?
I think it's going to be helpful. I think at the moment it's wonderful that that sequencing can be done more and more cheaply and more information obtained to really come up with precision medicine, as it's called. It used to be personalized, but it's become precision. And I think, though, that most of the targets that we're studying, because of the lag between discovery and application, most of the targets that we're studying were identified by more traditional means.
Yeah, the access to the genome gives us a huge technological advantage in looking precisely at what we're doing. I'm not sure it's been a trigger in discovering targets. I mean, it helped, of course, but it's not something that has been unlocked and then you can. It's not exactly the way it happened. Those were parallel efforts, but obviously, now the power of high throughput sequencing, deep sequencing, and all is very important for what we're doing. So those are tools that are essential to what we're doing now.
And Olivier, you mentioned some of the technological advances, and I think one of the key hurdles, especially in gene therapy, has been delivery. So what are the breakthroughs that have occurred there that have really gotten us over the hurdle? And maybe where do you even see delivery ultimately going in gene therapy in particular?
So we've made a lot of progress. I mean, on delivery, and delivery. I think this is the essential problem. This is what we're talking about all day, right? How do we improve delivery? How do we make it specific, safe, etc. So, as Malcolm was saying, it's been a slow progress. I mean, a continuous progress over the past 20 years, improving the systems, the vector system for gene transfer, and the systems for handling cells ex vivo, reimplanting these cells. And this has now led to two technologies which are robust enough that they can be really, taken over by the industry, by the healthcare, and applied to patients. So we hear a lot about AAV vectors, for instance, which is something that started probably 20 years ago, had a slow start, and then over the past 10 years really boomed a lot. And it's a vector system where suddenly you can do things that you wouldn't have imagined 10 years ago, right? You can inject vector into the circulation of a patient and get gene transfer pretty much everywhere in the muscle system, for instance. So immediately there's a possibility for addressing needs like muscular dystrophies, which were totally unmet and still are unmet, right? Yeah. But there is now some light, and you can go there. So there's a lot of excitement about that. Of course, there are safety issues, there are stability issues, there are all kinds of issues that, but we think, many people think that it's worth going and looking after that. What I'm trying to say here is that the delivery problem is not solved, but it's a problem that looks like it can be solved, it can be improved, and it's not only the business of fundamental science trying to come up with this principle now, it's also the business of the industry and people developing this as products for patients.
And Malcolm, I don't mean to classify you as cells and genes, because I know there's a lot of overlap between the two. But delivery can also apply to cell therapy and some of the stuff that you're doing. And I think one of the very interesting things that sometimes people forget, and actually Bob Peretti's talked to me about this multiple times, but with respect to cell therapy, we're really building on decades of stem cell transplantation effort and expertise. So we've really been doing this for a pretty long time. Excuse me. But what is happening and still needs to be figured out with respect to getting cells to the right place? Because we're kind of going to a different size and scale with respect to delivery of cells.
Yeah, that's a very critical question. And I think one of the reasons I got into gene therapy and cell therapy was I trained as an immunologist. My Ph.D. is in immunology, but I was interested in doing stem cell transplantation because it seemed to me that was an excellent way of delivering genes to the human body. And in principle, a single stem cell can repopulate a whole person for their entire life with genetically modified cells. And of course, one of the miracles of hematopoietic stem cell transplantation is that you inject all these stem cells into the peripheral blood. They have to go through various filters in the lung, the liver, the spleen, and yet somehow they lodge in the right place at the right time in the marrow, and there they grow, proliferate, and produce new progeny lifelong. So clearly it is possible to deliver cells and whatever they contain to the right place. The question is, how do you do it? And I think the answer is you try and adapt physiological approaches as much as possible. And it's very interesting that there's a great interest in T-cell CAR therapy. And I think that as we explore it further, for example, for the use of solid tumors, we'll discover that T cells alone are not the answer, that the immune system has many different components for a purpose, that each of them really recruits other components. And I think when we combine cell therapies, when we start studying how that can be done, then we'll really be able to deliver a much broader variety.
Well, it is really exciting, and even just 10 minutes into this conversation, you can really see that we're just at the tip of the iceberg with respect to these advanced therapies and technologies. We brought up safety. I think this is a really crucial topic to discuss. So Malcolm, I'll ask to stay with you. In most cases here, we're talking about, with respect to stem cell transplantation, obviously autologous cells or a patient's own cells. So that does alleviate some of the issues with respect to rejection and things like that. But there are still some pretty cumbersome conditioning regimens and processes that are involved, and even just the long-term potential consequences of introducing expanded or even modified cells to a person. So have we signaled the all clear on safety here with cell therapy, or is there still more work to be done and more to learn there?
Well, I remember the early days of cell and gene therapy when people used to present their phase 1 data and there were no benefits to the patients, but they'd say, oh, well, at least it was safe. And I'd always say, well, if something doesn't work, it's very easy for it to be safe. The problem is once you start intervening and once you start having potent agents, then you are inevitably going to have unwanted toxicities. It's never going to go away. So the difference, I think, between small molecule toxicities and cell therapies and gene therapy toxicities is that while the one tends to get better with time as the drug is eliminated, cell therapies, gene therapies can actually persist and worsen with time. And the classic example of that is graft versus host disease after stem cell transplantation. So yes, I think that control mechanisms, safety mechanisms are going to be crucial. Their absence will delay development, and their absence will increase the toxicity of many of these therapies down the road. But they are available and have been tested and shown to be extremely potent. So they're there, and we just are going to need to use them for many, many applications.
And Olivier, especially respectfully with gene therapy's rocky start, what are you guys doing at Biogen, and what is the field doing in general to make sure that these products are safe and do more good than harm?
Okay, so there are a number of improvements to be made in terms of specificity. We're talking about putting vectors in complex tissue systems, and we want to make sure we hit the right cells. So that's one area of research and development. The other very important thing is that I think we need to apply to cell and gene therapy, and everybody's doing that now, the standards of the industry, right? Years ago, it was made maybe a little bit more likely in purely academic environment, and the FDA was being, more gentle, probably. This obviously stopped because this led to problems, and the rigor of industrial development is something that's very important today. That's crucial. I think there's a number of things that cannot be done by academics. It's not their job. It's not their interest. So we have to marry the creativity of academia with the efficiency and rigor of what's happening in the industry. And that's part of all what we're seeing. This is also the shift, the sociological shift that Malcolm was referring to. Players, there's more and more players from the industry, and because it's there, now is the time for them. This is when and this is how we're going to improve safety. So, I mean, we could get into technical details of how, but basically, it is very much on the agenda, and this is and it could be. Those may not be simple engineering things, right? We may have to understand more about what we're doing. When we put in a vector, what's going on with the immune system? It's complicated, right? Nobody really has an answer. How do we go around this kind of problems? So that's what keeps us very busy.
Good. And obviously the promise of these advanced therapies is these step-ups in efficacy that we are starting to see. And, some people even uttering the word cure. I mean, really, really exciting advances. And, to me, this means both treating diseases where we haven't had any options before or any therapeutic alternatives, but also potentially increasing response of duration in very meaningful ways. So, Olivier, I'll come back to you on this one first. What do new gene therapies need to show in order to be considered effective with respect to, the treatment paradigm, some of the expenses, things like this? I know it's very indication specific, so it's tough to say, it has to show 20% or whatever, but it may be tough to generalize. But, what kind of dramatic improvements do we need to see? Does it have to be dramatic in a few patients? Does it have to be more patients that are seeing it? How do you think of efficacy with respect to gene therapies?
Well, those are still experimental treatments with sometimes safety issues, so risk. So you better show a big benefit, right? And of course, if your medical need is totally unmet, this is actually where we're going to start to look at the possibilities of developing these therapies. This has always been the approach, actually, because it makes sense, even with some risk, to intervene with gene therapy. This is what's been done with the the genetic immunodeficiencies in the early days, which were the first real successes of gene therapy, that came with safety issues and toxicities, right? But even if you take this into consideration, this toxicity, these adverse events, those treatments are still much better than anything that was proposed to these patients. So, I would still call it a success, right? So we need, to go back to your question, I think we need to be, to have pretty high standards on what we want to achieve, right? It's not about having a couple of month improvement in, of more life in a cancer patient. It's something that has to be sometimes much more radical, usually much more radical, because we need to show that these technologies can really make a difference in situations where either nothing exists or the current treatments are not sufficient, but then you need to show it's not just another pill. It's something which is radically different, has to be established with a very strong basis of success and demonstration of efficacy.
And Malcolm, CAR Ts and regenerative medicines promise to treat diseases in fundamentally different ways. But what do we need to see from this area? Is it similar to what Olivier is talking about? And how do you see product improvements or enhanced iterations even being developed from these products?
So I think Olivier is absolutely right. You've got in all these settings to see big improvements in serious diseases, marginal temporary improvements and not going to cut it. I remember reading a paper in Nature Medicine about using gene therapy to treat lactose intolerance in mice. And no one's going to give gene therapy to treat flatulence. It's not going to happen. So what we have to look for is something where we can actually make a difference to a disease that is otherwise untreatable and measure that difference in small-scale studies. And I think that's the critical thing, to improve the quality of our early phase studies so that we can actually measure improvements, even if at that time they are not cures. Because as you say, a lot of these phase 1 studies are in fact going to have to be iterative. There are many components in a complex biologic or advanced therapeutic, and it may be necessary to tweak them before they become optimized. But if you can measure some benefit, some of the effects that you desire in the first 1 or 2 or 3 patients, then you can go to a second iterative study and very rapidly build up a portfolio of components.
That's really helpful so far. So as I promised, I do want to keep it interactive. So with that kind of general couple first questions, if anyone has any questions, I'd love to bring the audience into the discussion. But I also want to ask both Malcolm and Olivier some of the specifics about what you guys are working on, because this really is cutting edge, super cool stuff. So Malcolm, what are some of the things that you're working on at the Center for Cell and Gene Therapy that really have you excited right now?
I think really where our main focus now is on taking advantage of one of the major properties of cells. You talked about delivery, and that's true. That's important. But above that, what T cells can do, for example, is recognize patterns. And I think it's going to be very difficult to come up with a single target to distinguish a malignant solid tumor from a non-malignant tissue. I think many of the target antigens are going to be shared between normal tissues or not going to be expressed by all the tumor cells and so on. But what I think you can do with a T cell is put into it receptors that will allow it to exercise pattern recognition by so-called Boolean logic, that is to have and or not receptors. In other words, it will only recognize a target if it has X and Y or Z, but not A. And that means that you can train it to look for antigens that are present on the tumor, absent on normal tissue. You can train it to look for environmental cues that will trigger the T cell when it reaches the tumor, but will keep it inert when it's in normal tissue, and so on. And so far, we have developed a lot of different approaches for doing that, and have a lot of components that will provide those kind of logic gates. And I think that's really where the interest for solid tumors will be most productive. We're also interested in using cells to deliver vectors, like Olivier says.
Very, very neat. Olivier, what do you guys, I mean, I know you have some programs, and again, congratulations on the very exciting data on Friday for Alzheimer's disease. That was fantastic as an institution, and I think for the industry. I know you have some things that you've disclosed in terms of gene therapy efforts at Biogen, but what are some of the other big areas that you guys are thinking about as well?
Well, we're thinking very broadly because there are many different therapeutic areas where gene therapy could be applied and where you have true unmet needs that we believe can be addressed reasonably well today. So it means that, apart from our interest at Biogen for neurology, hematology, and immunology, we're also looking at systems where, pretty much a lot of people have been interested in, like the eye. Ophthalmology is an area where gene therapy can really make a difference. We're looking at, I was talking about the muscle disease. This is also something that we're carefully looking at. And of course, the CNS is but for the time being, although we have exploratory, generally we consider gene therapy as a novel therapeutic modality that we can plug in different therapeutic areas that we have. So you're talking about Alzheimer. We don't truly have a gene therapy activity around Alzheimer, but we're definitely thinking around that, thinking of how can we use these technologies to address these problems, knowing very well that this is a very long shot, and when we're doing that, we're exploring, we're not developing products. But Biogen is a highly interesting place to precisely do that in targets like Alzheimer, Parkinson, the motor neuron degenerations, for instance.
Excellent. And one thing too, just even from having this conversation that's kind of jumped into my mind is, we have made advances, and this is kind of grossly speaking, from surgery to radiation to chemo to targeted therapies, and now to, T cells or immuno-oncology and gene therapy. So how do these new advanced regenerative and cell and gene therapies play in the sandbox with existing standards of care? In some cases, do they just surpass them? Do they get used in combination? I know that's kind of a very overly general statement, but how do you start to think about that? And I know you guys are more on the discovery side, a little bit less on the development side, but any initial thoughts might be really interesting.
I think they'll begin as complementary, as an additional option, and I think gradually they'll replace most of the conventional therapies. That would be the hope. And of course, there's a whole medical industrial complex built around, particularly for cancer, built around conventional therapies. There are whole institutions that make a living out of treating the consequences of surgery, radiation, and chemotherapy. And that kind of funding will be available to pay for these more advanced therapies. But it'll be a gradual process, obviously.
Bob, did you have a question?
I did. Malcolm. Thank you. First of all, great discussion, gentlemen. Thank you. It's fantastic. I'll start with Malcolm and Olivier. I'd like you to address this as well. But it strikes me when you were talking about the ability to train the T cells, that's a big evolution from where we are. And I hadn't really thought of it in this way, but up to this point with cell therapies, we've been relinquishing activity to the cell because we know that the medicines we have don't work particularly well. So we've been giving it to the cell, saying, well, if we can't figure it out, maybe the cell can.
Do it.
But that's made it hard to characterize the products and to release them and have rational drug design around really firm mechanisms of action and the like. So is this one of the reasons that we're moving into an inflection point here, is that now we're able to define the mechanism of action in a way, actually tailor and make it happen, so we can better test whether we're creating the right products?
Yeah, I think that's helping. I think a lot of that's definitely a component of why things have got better. But I still believe that the main interest really from the sort of inside or outside viewpoint, I think people started getting very interested when two things happened with the PD-1 and CTLA-4 Ig antibodies came along, and they realized that that was working not as every other monoclonal antibody had done by targeting something on a cancer, but by boosting the immune system. And second, the CD19 CAR studies on leukemia. And I think people suddenly realized, wow, these really can work. So I think that was probably the inflection point. But leading up to it and giving a justified basis for this emotional, sociological realization is a lot of other work, including the type that you described.
You think it'll make it easier for a company like Biogen to apply big pharma principles of rational drug design now that we better understand?
I'm not sure I understand your question. Are you asking whether it's easier for...
Yeah, so one of the challenges that we've had is really understanding the products, right? So we put the cells in the body and we kind of figure we know what they're doing. We make up putative mechanisms of action and work around that. And from my view, which comes from the manufacturing and the building the product side, it's a challenge, right, for product characterization and for developing products around a spine that is difficult to define. So.
No, it is a challenge, and we have to educate the different experts within a company like Biogen at the specific problems of cell or gene therapy. How do you define a dose? What is target engagement? What is, I mean, all these things that a company like Biogen and others do very well. How do we adapt that to cell and gene therapy? I don't think the answer is obvious all the time. I mean, there are answers, and there's a lot of enthusiasm in finding answers, but what I'm saying is that a lot of this work in putting gene and cell therapy out there to patients needs to go through that process of understanding how you actually develop a product and how you transform your ways into, you have to do new things for sure. Those are ideally one-time treatment often. So how do you design your toxicology studies in this case? How do you commercialize it? I think that was one of the points you also wanted to discuss, right? So everything is kind of challenged. All the position in the chain of development and commercialization in the company are challenged by this advent of new novel ways of doing things.
Thank you.
It is really interesting because the future that you guys are proposing, already things have moved so quickly over the last few years, but you think about, and I'm not picking on Pfizer per se, but you think about the big pharma companies today, and they are not at all positioned for this future of medicine that you're talking about. So it's really interesting. There's going to be a lot of big changes, a lot of big investor opportunities within that. Olivier, you start to mention commercialization and scale up. And again, I know that this is even yet another step away from where we are now, but how do we start to think about advanced therapies, cellular therapies, or gene therapies in a scale-up condition and ultimately in commercializing these types of therapies?
I mean,
do we not have to worry about that yet, and let's prove the concept yet, and we'll worry about that as we get closer, or how should we start to think about that?
We worry about that today, and the industry has taken challenges like, how do you bring monoclonal antibodies to patients, and it hasn't been easy.
They had that, yeah.
It's taken a long time. I mean, it's just now coming to fruition, really. So I see that as the same process, basically done with the same people, with the same expertise. A gene transfer vector is a biologics, maybe a little bit more complicated but the same people with this great expertise at bringing recombinant proteins to the market are tremendously interested and tremendously capable, I think, of doing it with gene transfer vectors. For someone coming mostly from the academia like myself, this is the attraction in working with a company like Biogen, because I find solutions for things that are definitely and expertise that I would not find in the academia. So I think this is the way it's going to happen. Now you were talking about a much bigger structure. I think the advantage of smaller big biotechs like Biogen, Amgen, Genzyme, is that if they want, they can fully integrate gene therapy into their operation. And not develop gene therapy as a little business unit on the side that you put some money and see how it grows and maybe you take it over or something like that. No, it has to be a real commitment that then, and everyone at every level of the company, in every function, feels that they have some job to do. Right?
In the DNA. Malcolm, how about your work at the hospital? How do you sort of foresee the discoveries that you're making? Where's the handoff for you guys? We've seen some really exciting academic collaborations with big pharma, with industry. What are your guys' view of how to do that?
The short answer is I don't know, and I think it's going to be different for each product. We've done it in several different ways. So the earliest kind of cell therapy we had was Epstein-Barr virus-specific T cells that we used to treat lymphomas and nasopharyngeal cancer. And the way we handed that off was after the phase 2 studies, which were very promising. So those have been licensed to 2 different biotech companies. We have a collaboration with Celgene in which we licensed a whole area, CAR T cells, for them to develop with Bluebird Bio. So that's a much broader and much earlier phase of development. For the safety switches, we licensed that after our phase 1 study to Bellicum. And then we also have people in-house who are trying to develop their own startup companies with their own technologies. So all 3 ways.
What about the regulatory agency? Because they're kind of the goalkeeper or the gating agency, however we want to think of them. So ultimately getting these advanced therapies to product. Where are they? Where is their bandwidth? Where is their mind frame? Where's their understanding about some of these advanced therapies that are coming down the pipe?
I think we come from a situation maybe 15 years ago where there was almost no expertise in the regulatory agency on these topics, and it's been a great learning curve. And personally, I've always found the interaction I had, and actually Malcolm probably had more interaction than I do, but he can testify, as very productive because these people really wanted to learn about it and actually build the regulatory framework along with us. So it was not, this is the book, you have to go by that. I mean, there's some level of that, of course, and we all agree on that. But as I was saying, for any other things, you have to define a number of things differently for gene or cell therapy. You have, there's just a definition of the glossary is different. Yeah. So you have to agree on that. Then you have to educate them on the technology and what we want to do. And they're reasonably smart people. They can learn things. And I've always found that there was a real proactive attitude in Europe and also over here in the United States, but we're still in a learning curve, I would say. But I don't know. I don't see any real negative aspects of that, yeah, of the process as it is.
Malcolm, what are your views on that?
I obviously can't speak for the agency, but again, from the outside and dealing with them, I would say 2 things have happened. The first is that as efficacy has gone up, they're much better able to judge the risk-benefit ratios, which is always very important for any regulatory agency, obviously. And I would also say that they're a little bit disappointed that we're all still following the conventional phase 1, phase 2, phase 3 route. For example, the phase 1 studies in cancer and many other diseases are not intended to show benefit. They're intended to be dose finding with dose escalation to the maximum tolerated dose. Well, for most cell therapies in particular, and for some gene therapies, there is no true maximum tolerated dose. What there is a threshold dose below which very little happens and above which events happen. And the difference in dose only affects the time at which the event occurs by 1 or 2 days, because, of course, the cells generally proliferate. So they would, I think, much prefer us to develop flexible phase 1s in which we modify the components after 2 or 3 patients, and we look in a very data-intense and very accurate way at the consequences for the patient of each treatment. In that way, we can modify the phase 1 as it's in progress and really come up with some efficacy data as well. So kind of phase 1/phase 2 without this obsession with finding a maximum tolerated dose.
Yeah, I think those are really good points. And I had to add, I think maybe the agency is the unsung hero in this biotech bull market, because without them, we really couldn't see the progress that's being made. And I don't think they often get the recognition that they deserve in terms of the work that they do and the progress that has been made. So I want to see, we're starting to get close to our time. Are there any questions? I don't think we can possibly have a discussion about advanced therapies without talking about pricing a little bit. So I'll save kind of my doozy of a question for last and ask about reimbursement. How do we afford, how do we pay for, how should we think about pricing and the value proposition of these drugs?
We've thought about this a lot, and I think one of the ways that they're going to be priced is as replacements. They're not add-ons. They're going to be instead of a lot of expensive therapies. A bone marrow transplant can cost anywhere from $100,000 to well over $1 million. If you could replace that with a cell therapy, that would be that was less inconvenient. That would be a major plus. I think there's also going to be possibilities of paying as a kind of reverse annuity. So the longer the patient survives, you pay a certain amount each year. Insurance companies may be willing to consider that. But I think in the end, ultimately, it's going to be showing that the cost-benefit ratio is positive. And what that means is that along with having phase 1 studies that are designed to show efficacy, we're going to need to get health services research people involved very early on, I think, and start costing what these therapies actually are, what their cost actually is, and what benefits they produce, and what savings they produce. And I think instead of waiting till post-marketing studies or whatever to do those cost-effectiveness analyses, they need to be done early on. They may not produce the answer you want, but then at least you know before you try and sell something that no one will pay for.
Olivier, anything to add?
Yeah, no, I don't have much to add to that, but I just want to stress the point that obviously this is going to be looked at in terms of efficacy, unmet need. So again, back to the discussion we had before, this is where it's very important to show that you make a difference. If you make a difference, I don't think there'll be much of a problem. There'll be adjustments to be made. You know, how do you really pay? Do you have some kind of mortgage or some? But for sure, it's not. I mean, nothing's going to happen if it's only a cosmetic type of intervention. So, I mean, I know people are working hard at figuring out models and how it could be done. I don't think anybody really have a. We'll need a true life situation, right? To really see.
We'll get one soon with
We'll get a couple soon. So we'll be more advanced in even in a year from now, trying to figure out.
So with our last 2 minutes, I have to be honest. I have been shocked at just how much has happened in the last 12 months. I knew things were happening fast, and I knew, I get to sit and be a pretty close observer, maybe have a front row seat to everything going on to this, but between what's happened in the capital markets, what's happening in clinical studies, I think I've been even more surprised by how quickly things are moving than I would have even anticipated sitting on this stage last year. So what are your surprise predictions for the next 12 months? What is going to blow us away? And it doesn't even have to be 12 months, but what's your surprise prediction for the future? What do you think is going to really surprise people even more than where we are today? That's an unfair question.
Yeah, I didn't really think of this one, but
I know. I think you've confused me with sort of seer. Yeah, no.
I think that again, it's about things that would be unimaginable 5 or 10 years ago. Like, can you really modify enough cell in a brain of a patient with a genetic neurodegeneration of metabolic origin, for instance, to counteract that? And what we're seeing in animal models today is that, yes, we probably can. There are issues, is it safe enough? But those are terrible diseases. And I think we could be astonished by, the result of trials which are now starting, where you inject a vector in the circulation of a patient whose brain is rapidly going away, and you you stop the degeneration, and maybe actually you just recover some function, right?
I
So
think
this
that
is, I mean,
counts.
again, you ask for prediction or wild dreams, but I think this is not too far. And those are things that you would have asked me that 5 years ago. I would have told you, what are you
No
talking
way.
about?
Yep.
So that's where we.
And Malcolm, you maybe already gave us one with that, multi-receptor T cell thing was pretty far out there and pretty cool for me. So I'll let you off the hook with that one, unless you have something else.
Well,
that you
I
come up
think
with.
what we'll be seeing over the next 12 months is people beginning to integrate all the different components, integrating oncolytic viruses, T cells that recognize those viruses and tumor, and antibodies that enhance the immune response. I think integration of the components, that's the where the future is going to be over the next couple of years.
Excellent. Well, gentlemen, thank you sincerely for your time today. This is really, really exciting, really, really worthwhile. So thank you for joining us.
Thank you, Ted.
Thank you.